Hello everyone and welcome back to Recipes for Runts. This is Joe. Today I'm going to be doing a step-by-step video on how to make a plant cell cake. So let's get to it. Now, as most of you already know, cells are made up of organelles. And because a lot of organelles just go into one tiny microscopic cell, I'm going to need a lot of stuff to make them. So for this project, I'm going to be using a decadent chocolate cake. I made chocolate cake in a 9 by 13 inch pan, and then I cut off all of the corners to make it look like a hexagon. Then I'm using a red edible ink marker for the chromatin, green icing with a star tip for the chloroplasts, blue icing in a plastic bag for the vacuole, chocolate fudge mica for the mitochondria, dark green fondant for the cell wall, compound pink chocolate for the Golgi apparatus, light green fondant for the cell membrane, some white fondant which I will color red and orange for the nucleus, some small white sugar pearls for the ribosomes, brown sugar which I'll use for the endoplasmic reticulum, and lime green icing to stick everything together and also for the cytoplasm. And these are all the things I'm going to be using for my cell cake. So the first thing I'm going to do is I'm going to take my chocolate cake and give it a crumb coat using the lime green icing. What this does is it catches all of the chocolate crumbs, so when we put on another layer of frosting, it'll be nice and smooth and crumb-free. Now I'm cutting off a small portion of my white fondant, and I'm going to dye a small amount of it red. And then I'm going to dye the rest of the white fondant a light orange. Then I'm using icing sugar. You can use cornstarch or whatever you want just to stop the fondant from sticking, and then rolling it out. And then for the nucleus, I took the leftover cake scraps, crumbled them up and mixed in some icing, and then I formed it into a sort of semi-sphere shape and then cut off half of it, leaving a little bit at the bottom. Then I'm carefully covering it with the orange fondant. Make sure to keep on smoothing and cutting with a sharp paring knife to have as little creases as possible. Then using a wet pastry brush, I gently brush off the icing sugar to leave it clean. Then I'm rolling my red fondant into a ball, which I'm going to use for the nucleolus. Then I cut off sections of it so it can fit snugly into my nucleus. Then put it in the fridge to chill while you do the next step. Then take your cake out of the fridge and using your offset spatula once again, put a smooth layer of frosting over the entire cake. And as you can see, the second coat of frosting is clean and crumb-free. If we didn't crumb coat the cake in the first place, then the crumbs would be contaminating the second coat of frosting. I'm using an icing comb to smooth off the top and the sides of the cake. The sides don't need to be perfect, but make sure the top is as good as you can get it to be. Then, you guessed it, put it back in the fridge while you do the next step. Now take your nucleus out of the fridge, making sure that it's completely dry, and then using the red marker, draw some strands of chromatin. Now for the cell wall, I'm actually going to be mixing together my two different greens, but make sure you leave a little bit of light green left over for the cell membrane. Then keep on kneading the fondant like bread, adding more icing sugar to keep it from sticking until it's all mixed together. Here is the result. Set the green fondant aside, and then take out your brown sugar. You can use light or dark, whatever you want, and then take out a half a cup and put it in a small bowl. Then add a couple of tablespoons of water and a half a tablespoon of cinnamon. Then mix it together, adding more water or brown sugar if necessary. Now this is the tricky part. You want it to be the correct consistency. So when you drip some off your spoon, it shouldn't run like water or fall down and glop. It should just start to ooze back down. And you also want to make sure there's no more lumps of brown sugar or cinnamon so they don't get stuck in our piping bag when we're piping it. Then put it in a piping bag, cut out a small hole about 1/3 of a centimeter thick, and then set it aside. Now for the cell wall. Take some of the green fondant that you made earlier and roll it out into a long snake. When you finished rolling, flatten it out to 1/2 centimeter thick. Then take a small piece of your fondant and then you're going to cut it out. Using a tape measure really helps. And you want the height of the piece to be the same height as the cake. You can make it a centimeter taller if you would like to as well. Just do whatever you want. Then using several pieces, cover the sides of your cake completely. This is what it should look like when it's done. Brush off all the excess icing sugar using a pastry brush. Then using the rest of the green fondant, make small, thick, rectangular shapes the same height as the cake and place them on the corners. And as you can already see, it's starting to take up the shape of a plant cell. Continue adding more around the wall. You can do all of them on the corners if you want, or on the sides like I'm doing here. You can do it randomly or uneven or in a pattern. It's really up to you. Our cake and cell wall is fully assembled, and now we're ready to start adding the organelles. Use a small dollop of icing to glue down your nucleus. Then take out your brown sugar cinnamon mixture and make little squiggly lines around the nucleus for the endoplasmic reticulum. Then pipe out the vacuole anywhere on the cake using the blue icing and spread it all in so it looks neat. And I'm also using a little bit of white icing to add a reflection in the vacuole. You can skip this part if you'd like. Then add on your green icing in small green pods for the chloroplasts, and then your chocolate fudge micanics for the mitochondria. Then randomly place a bunch of sugar pearls on the cake for the ribosomes. Now for the Golgi apparatus. I took about a quarter cup of those pink chocolate melts that I showed you at the beginning, and I melted them in 20-second intervals in the microwave just until it was nice and smooth as shown here. Then I put it into a plastic sandwich bag. You can also use a piping bag if you'd like. Then I placed down a small sheet of parchment paper, and I used some objects just to weight it down so it didn't curl, and then I carefully piped out the Golgi apparatus. You don't have to use compound chocolate like I'm using here. You could use fondant or other candies if you'd like. And when finished piping, put it in the fridge for about 5 minutes to set. Now roll out your leftover light green fondant into a really thin snake and then place it all the way around the cake for the cell membrane. If you need to, use a little bit of water to stick it down. After 5 minutes, our Golgi apparatus should be nice and set, and then just place it onto the cake. And finally, I sprayed on a little bit of edible green mist spray to give the cake a nice shading and to make it look a bit more 3D. And there you go. There's the finished plant cell cake. And here are some pictures of the end result. Later on, I put on some edible white chocolate rocks for the lysosomes. I'm very sorry I couldn't show that because I didn't videotape it. But anyway, I thought it still came out really good and pretty darn close to what an actual plant cell looks like. Special shout out to my amazing friends and science teacher too. So thank you all very much for watching, and be sure to subscribe to Recipes for Runts. See you guys!